All right, it's a minute after, so I think we should get started. So welcome to the Director's Seminar. This is, I think, one more before the end of the semester, so thanks for joining us. Today, I will have the pleasure of introducing Professor Gabriele Gratton, and he's going to talk about his research. I'm Jeff Moore, and it's really been my pleasure to bring together the group of faculty members from the Beckman Institute who really are doing exciting, barrier-breaking research and give a chance to present their work in a format that I think is accessible to almost everyone. So we try to make sure that the talks are directed to the broad audience that exists here at the Beckman Institute. Our format for question and answers will be for you to put anything in the chat feature that you would like, the Zoom chat feature. And at the end, Gabriele will respond to those questions as time permits. Let me introduce Professor Gratton. Gabriele is a professor of psychology here at the University of Illinois, and he has been a full-time faculty member since he began on our campus. He's a member of Beckman's Cognitive Neuroscience Group. He received his MD from the University of Rome and his PhD in psychology from the University of Illinois. Along the way, he's received several distinctions during his career, including an award for early career contribution and subsequently a Lifetime Achievement Award from the Society of Psychophysiological Research. Gabriele's research applies neuroimaging and electrophysiological technologies to better understand brain function. In particular, he's interested in studying the organization of cognitive processes with attention to the effects of aging. In today's seminar, I think you're going to hear Gabriele's going to discuss diffuse optical imaging, which is a non-invasive tool that's used to study deep tissues in the brain. He's going to focus on the types of functional brain measures that can be obtained with this technique and how that allows researchers to analyze the spread of neural activity during the processing of external stimuli and in preparation, for example, for motor activity. He'll also discuss the relationship between these measures and various psychological processes like language processing, sensory and working memory, task switching, and cognitive control. As you can see, his presentation is diffuse optical imaging of the human brain. Gabriele, thanks for joining us today.
Thanks, Jeff, for the very kind introduction. So today I will be talking about diffuse optical imaging. And to start with, I will talk about the fact that optical imaging is a very common and very basic modality in neuroscience. It can be used to study many different things. In fact, at Beckman, there are several laboratories that do various forms of optical imaging. However, in non-invasive human studies, the situation is much more complex because, of course, the surface tissue, which includes skin, skull, and dura, hides the brain, making it difficult to use wide field illumination to generate images. And today I will be talking about the work that we do in our lab on the diffuse optical tomography, which is a tool that provides a partial solution to this problem, and with main limitation associated with limited penetration and lower spatial resolution. So first of all, let's understand why we cannot see directly through the brain in a normal human brain, in a normal person. So you all over this moment will have light arriving to the top of your head, and some of these photons that are in the light will be reflected back at the surface of the head. Some of them, however, will penetrate through the head. That depends in part on the incidence angle that these photons have. And when they actually penetrate, they enter into a different medium. So when this medium that we're talking about, the human head, as most of the brain of the tissues in the human body, are very highly scattering medium. So what happens is that the photons that penetrate through the medium begin a process of scattering process in which they are basically diffused through the medium in a very random fashion. Some of these photons ends also being absorbed by the tissue itself, because that will partly depend on which type of molecules they will hit and what type of light we are going to use. In addition, some of these scattered neurons, they will migrate to all of the brain, and then they will come back to the surface, and they will re-emerge out of the surface. This last set of photons are the one that we are actually measuring when we are doing our technology. Of course, as I said before, these two processes, absorption and scattering, are two most very important processes that impede our way of seeing the brain from outside. One aspect of this is that absorption is largely determined by a few substances that are highly concentrated and have strong absorption spectra. Of these, of course, one is water, which, however, tend to have a low absorption between 300 and 1000 nanometers. Most importantly, also, there are also oxy and deoxyhemoglobin, that are contained in the red cells in the blood, and that absorb light in the visible spectrum. So this means that to have a good amount of light going to be scattered and then re-emerge on the brain for our measurements, we actually need to use wavelengths at which the absorption of hemoglobin is reduced. And in particular, this happens between wavelength around between 700 and 1000 nanometers, a window which we call the near infrared window. In this window, however, the other process, the scattering process, becomes the most important reasons for most important obstacles to the movement of the photons and through the brain. But it's not only an obstacle, it's also the supporting system by which light is actually moved to the brain. But so in this sense, however, there is a new phenomenon, which is basically what we call a scattering dominant situation, in which we have a diffuse type of regime. And as I said, the window for us is in the near infrared light. Now, the combinations, the basic setup that we use in our lab and in most of the diffuse optical imaging lab is that having sources of light that illuminate a portion of the head, and then have a detector which is located at some distance from this source. By using this type of approach in which we separate sources and detectors, we can reduce the influence of this immediately refracted light, or immediately backscattered light in the first millimeter or few millimeters of the tissue, and instead be able to see photons that travel deeply into the tissue before reaching the detectors. Notice that these photons are actually moving in a random fashion. So of all the photons that are produced to a source, we only pick up a portion of them. Those that end up coming out from the tissue at the point where we have a detector. And this combination of a source and a detector identify a probabilistic volume in which the photons might have traveled. And all the measurements that we can do about the movement, the transference of photons between the source and the detector, will refer to what happens in this particular volume, which sometimes is called with a very technical term, a banana. Now, of course, we may do this with a single channel, or we can do it a lot of sources and detectors, and do recordings from a lot of places at the same time in parallel, or using multiplexing methods. And in this particular way, we can measure activity from a large number of pairs of sources and detectors. We can also co-register these sources and detectors with images of the brain, like those that can be obtained with a structural MRI method, and they use this to refer the visual measures to particular areas of the brain. Even more, we can also model the way in which photons will likely travel between the source and the detector. So, and use these models to generate forward models about the transfer of these photons and backward models about how activity in the brain may influence these activities. And so these are all parts of the procedure that we use to reconstruct where in the brain we actually see activity using this particular approach. Now, another aspect of the near-infrared light is that even though the window of light that we are using is only between 700 and 8,000 nanometers, still there are differences in the absorption spectra of different substances within this particular interval. In particular, one important distinction that has been made in the field of diffuse optical imaging is between the absorption spectra of oxy and deoxyhemoglobin. So the hemoglobin, which is bound to oxygen, or that is just without the oxygen. These two species of hemoglobin have actually different types of absorption spectra. And we can employ this by using a spectroscopic approach in which we have light of multiple wavelength, like say 690 and 830 nanometers, and use these to actually estimate concentrations of oxy and deoxyhemoglobin present in tissue as a function of by using different applications of what is called the Beer-Lambert law. Now, light transmission through the brain, of course, if you put a source, declines with distance from the source. And this is due to both the tissue absorption and the scattering. And in thick diffuse medium, thick means that where scattering becomes a random process. And these thick media, the light transmitted between sources and detector is given by the combined effect of three parameters: the source detector distance and the absorption and scattering coefficients of the medium. Of course, each tissue and each part of the brain might have a different scattering and absorption coefficient, but you can also consider the cumulated effect of all of them across different areas. There is a particular quantity, which we call the EAC, or effective attenuation coefficients, which is actually given by the rate of decline of the light transmission through tissue as a function of the source detector distance. And as is shown in this formula, this parameter, effective attenuation coefficients, is related to the harmonic mean of the absorption and scattering coefficients of the tissue itself. Now, it is also the case that this parameter, the effective attenuation coefficients, varies both in terms of locations, but also in terms of individuals. And there are age-related variations in these parameters that are likely related to changes in important phenomena for people that study aging, like tissue oxygenation and cortical thinning. So this figure indicates a results we obtained from an experiment in our lab, where the average value of this EAC parameter is estimated for a bunch of individuals varying in age between 18 and 78 years old. Also, different estimates are obtained with light used at 600 nanometers and light at 830 nanometers. Using this approach, you can see that there is a common response across different wavelengths. And these common variations in this parameter is interpreted as due to changes in scattering. And in aging, we know that the scattering of the brain can change because the atrophy, cortical atrophy, can lead to and also atrophy of the rest of the brain can lead to an increase in the cerebrospinal fluid in the subarachnoid space, which is low in scattering. And a consequence of this is that the head becomes more transparent. And in fact, actually, these EAC measures obtained across the whole head is strongly correlated with cortical thickness in this sample of subjects, which varies with age, but it will be correlated with cortical thickness even with partial out age. Also, we can see a differential effect. So some individuals, even with the same, have more effects at 690 and some have more effects at 830, or different rates of these two phenomena, of this EAC and the two measures. These differential measures that we have at these two wavelengths is largely due to the fact that there is different absorption at different wavelengths, and that is due to oxy and deoxyhemoglobin. And the consequence of this is that we can also use this type of measure to study brain tissue oxygenations. And in fact, in this study, in the same subjects, we can show that there is a correlation of this estimate of tissue oxygenation obtained in this fashion with actually with age, in which tissue oxygenation goes down with age. For the rest of my talk, I will be presenting a way in which we apply diffuse optical imaging to study dynamic phenomena in the brain, things that vary over time in the brain, even in a rapid fashion. And this is possible because, of course, the measures we have can be taken very, very rapidly, every few milliseconds. As a consequence, we can study different phenomena in the brain. And these phenomena will be related to different compartments of the brain itself that all together operates and perform important operations in the brain. So a leading idea behind this is that the way in which the human brain works, it requires the appropriate functioning of a number of different subsystems. One of the systems, of course, is the vascular system, in which arteries bring blood to the brain, and then the brain is diffused to capillaries. We also have a venous return, and so on and so forth. But in addition, this vascular system is influenced and also modifies effect activity in neural function. So there is a sort of a two-way interaction between the vascular system and the brain. And these two needs to be considered as a sort of a combined phenomenon that works together to perform activities. And of course, there are different types of brain function. We make neural functions that we may consider, like neuronal activity, oscillations in brain activity. And altogether, all of these are related to, can be related to brain function. Notice that we can produce separate measures using our approach for all of these fourth style of different phenomena that we have. We can have measures of the arterial function with a technology called pulse dot, measures of capillaries and tissue oxygenation and so on, which is contained with a technology called functional near-infrared spectroscopy, a method for studying neuronal activity, which we call EROS, if it is indicated for the synchronous time course activity or fast optical signals in general. So all of these measures are possible using the same approach, and in fact can be measured exactly at the same time. So all these compartments, according to us and to our data, contribute to cognition and behavior. In this sense, of course, let's examine one by one these four types of compartments and how we measure them. So first of all, let's talk about the arterial system and this technology, which we call pulse dot. So the time scale of the mesh phenomena that we are talking about are related to the cycle, the pulse cycle that occurs in the brain, which is of the order of 600 to 1500 milliseconds in duration. This is between a systole of the heart to the next systole of the heart. And then we have, of course, another phenomenon which is associated with this, which we call cerebrovascular reactivity, and I will talk about this in a minute. And then there are long-term effects that may accumulate over a long period of time. Now, our measurements are based on the measurement of the arterial pulse, which occurs very rapidly, but it produces strong changes in the amount of light that can move between a source and detector, or in light intensity. And these are related to difference in absorption by the tissue itself. So how is the arterial pulse generated? Well, the blood is brought we have to understand that the blood is brought to the brain by large and progressively branching elastic arteries running mostly on the surface of the brain. And these arteries branch into smaller muscular arteries and arterioles, which provide peripheral resistance and can contract and dilate, regulating blood flow to different brain areas. The pulsation occurs mostly in elastic arteries, but reflects the summation of both the forward wave coming from the heart, the systolic wave coming through, and the backward wave, which is generated by the peripheral resistance. So there is a forward wave that is transmitting through the arteries walls, and then there is a backward wave which comes back, coming from the tissue itself. And this is measured within the elastic arteries that are diffused on the surface of the head. Electric arteries, however, vary with age, and in particular, they become stiffer because of a loss of elastin. And these increase pulse wave velocity, the speed with which this pulse wave travels through the arteries. And this creates a different overlap between the forward and backward waves and change the shapes of the shape of the pulse. So you have in younger individuals with the more elastic arteries, the transmission is slower, the pulse moves slower through the arteries, and therefore you can separate the forward and backward wave and generate a complex wave with multiple peaks. If you instead, however, have older individuals in which the arteries are much less elastic, these the speed of transmission is very fast. In particular, the reflection coming back from the backward wave comes to overlap with the other waves that we're generating large, very peaky type of pulse waveforms. We can measure using this type of pulse shape. We're using optical methods with a technology which we call pulse dot. Pulse dot is similar logically to pulse oximetry, which is a similar optical techniques which is used to study the pulse in the finger or other peripheral part of the body. The main difference is that we are having a lot of sources and detectors, and we can measure it spatially across the head in all different areas and different locations. And of course, the measures, these optical measures, only refer to relatively surface areas because of the way in which light is introduced and measured. So in the diffuse optical approach. But still provides a good idea of where a lot of the surface areas where these arteries are moving, are placed around the head. So this particular idea is that all based on the idea that the fact that during a systole and diastole, there are different amounts of different pressures inside the arteries. And these distend the artery itself and create a different volume. And as a consequence, we can measure these changes using these particular parameters. And there are different parameters we can measure here. We can measure the amplitude of the pulse. We can measure the shape of the pulse, or we measure the delay that the pulse have with respect to, for instance, the R wave of the EKG. All of, in general, large amplitude, small prefix values, prefix is this shape measure, and short pulse transit times are indexes of arterial stiffness. And we have used this in a bunch of studies in the last, and we have a couple of grants, Monica and I, on this particular type of approach. And one of the main measures we are using is this prefix measures, which is a measure of pulse shape, which reflect more directly arterial elasticity in the tissue. Or in the arteries close to the tissue. And arterial elasticity decreases with age and increases with cardiorespiratory fitness. So in this case here, we have a prefix measure across different age and show a large reduction. Interestingly enough, in also the arterial elasticity, this prefix value tend to increase with cardiorespiratory fitness. That said, it is also the case that the measures of arterial of this prefix value tend to have different values in different areas of the brain, and in fact have not so only in different across subjects, but also in each particular individual. And one thing we can show is that these local changes in arterial elasticity, which are measured with prefix, tend to be correlated with local tissue loss or atrophy and white matter structural abnormalities. So this is the correlation with volume for these particular regions. All of these are standardized by the typical volume of that region and shows correlations between that increases as a function of age between prefix and the volume in a particular region. And this shows, this other plot on the right shows how prefix tend to decrease with, increase with, lower values of prefix are associated with white matter structural abnormalities, which is basically problems with your white matter in the brain. Optical measures can also be used not only in older adults, but also in preterm infants. And this problem exists in preterm infants, that their arteries are pretty brittle. That's because they contain still very little amount of elastin in their wall. And so the arteries tend to have a very high level of stiffness. And this problem of brittleness of the arteries causes difficulties regulating the blood flow to particular areas of the brain, in particular absorbing changes in blood pressure. And this may lead to intraventricular hemorrhage, which is a leading cause of cerebral palsy. Now, we have done studies with our collaborators in France on preterm infants and showed that, in fact, the specificity and specificity of pulse dot in detecting arterial brittleness and therefore the probability of intraventricular hemorrhages is higher than that obtained with other current standard methods to study arterial elasticity, like in the brain, like transcranial Doppler. Now, another aspect that is hinted by all of this is that, as I mentioned before, is that the pulse itself and the way in which arterial system works is not just passive. It is an active system in which we can actually react to particular conditions by changing the opening or closing of particular arterioles and the phenomenon which we call vasodilation or vasoconstriction. And this actually creates a reactivity in the cerebrovascular system. And so now these can be, there are many different ways in which we can induce cerebrovascular reactivity. One is changing blood in pressure, and like due to its head position, changes in oxygen and carbon dioxide concentration, which will be chemical signals. For instance, by having individuals do breath hold or breathing CO2 enriched air. And there also can be, and we talk about this later again, cerebrovascular activity, which is induced by neural activity itself. And this is basically the base for bold responses measured with functional MRI. Now, whenever we have inefficient cerebrovascular activity, we may have a decreased tissue oxygenation or alteration in this phenomenon. And so this can be a problematic situation. The cerebrovascular activity itself is actually much better in individuals with high cerebrovascular cardiopulmonary fitness. For instance, this is a measure of VO2 max and the correlation of the cerebrovascular reactivity with, in this case, measured with by breathing CO2 enriched air and the VO2 max. And you can see that there is a strong relationship between these two parameters. Now, that said, this so far I've talked about the arterial function. Then another thing we can study with diffuse optical imaging method are the functioning in the capillaries. And the functioning in the capillaries can be studied using a technology called functional near-infrared spectroscopy. In this case, what we are targeting is the amount of relative concentration of oxy and deoxyhemoglobin in the tissue that could vary as a function of task and as a function of neural activity or as a function of any other cerebrovascular activity with manipulation. These typically, we see two types of phenomena that influence this concentration of oxygen and deoxyhemoglobin. One is the consumption of oxygen, which occurs early on when during the if there is neural activity, but also this vasodilation and vasoconstriction, which is the reactivity itself. So these are a typical example of a curve of a concentration of oxygen and deoxyhemoglobin during a visual stimulation task. And you can see here that there is an increase in oxygen hemoglobin and a decrease in deoxygenated hemoglobin. These are measures taken from occipital cortex during a visual task. Now, cerebrovascular activity can be actually improved by using certain particular manipulations. We have talked before about cardiorespiratory fitness and physical exercise. Another type of manipulations that can improve the cerebrovascular activity are the ingestion of cocoflavanols. Cocoflavanols facilitate the production and release of nitric oxide by tissue. This nitric oxide is an important mediator of cerebrovascular activity. And their effect on cerebrovascular activity can be revealed by asking subjects that have taken this type of flavanols in a cocoa drink and compare that with the same measures obtained from people that have also drinking cocoa drink without having this flavanols in the cocoa. And the result shows that this is a controlled, double-blind, acute study, pre-post studies, comparing the effects of cocoa drinks with high and low flavanol concentration. And they show that the level of the type of response and the rapidity of the cerebrovascular response is improved very, very much by the ingestion of the flavanols with respect to what the other situation we have. Now, this study is based on normal young subjects, and even normal young subjects have an advantage. To tell the truth, not all of them have an advantage. In fact, people that have a very good cerebrovascular reactivity on their own don't seem to particularly profit from the flavanols, while individuals that don't actually get the best effect. Also, what we found in this study is that this is correlated with cognitive function. And so performing with subjects are asked to perform very difficult tasks, cognitive tasks, very complex, they actually improve clearly with the flavanols when they drink the flavanols the way they don't. Now, the other type of cerebrovascular activity we have been talking about is that associated with neural functions per se. And so this is also sometimes called neurovascular coupling. And neurovascular coupling is determined by the situation in which neurons are active. They consume energy or glucose, but they also produce some type of other type of chemicals in their activity that generate a set of responses. One aspect of this is, of course, is the consumption of oxygen, and which is required to metabolize glucose. This per se causes a drop in the concentration of oxyhemoglobin, but very rapidly the system reacts to this and generate by using various mechanisms, including the production of nitric oxide or acetylcholine by this system. They induce vasodilation, and inducing vasodilation, there is a big push of new blood coming from the heart and from the lungs that brings oxygen to the tissue through in the red cells, and this creates a large increase in oxyhemoglobin into the tissue itself. Now, this phenomenon of vasodilation and due to induced by neural activity is actually we call neurovascular coupling. And what we find is that in some cases, in older individuals, we have an impaired neurovascular coupling. So this is an example of a data we got from our lab showing this type of lack of response in occipital areas to particular neurovascular response in older adults. But in fact, we also find that this phenomenon in older adults is very different if we have older adults that have high in cardiorespiratory fitness or low in cardiorespiratory fitness. And so the large increase that we observe, in young individuals here is preserved in older individuals with high cardiorespiratory fitness, but is absent in older people with low cardiorespiratory fitness. That said, we can also map this brain activity in the brain, and we can show that if we have multiple sources and multiple detectors, like in an example like this, placed on occipital areas, we can actually generate 3 images of brain activity that is generated in this process. It ends up that the images we can obtain with the functional near-infrared spectroscopy, which is this technology based on changes in oxygen deoxyhemoglobin measured with optical methods, actually tends to overlap quite well with similar images obtained with bold signals in MRI. The main problem with these images is that we cannot observe very deep phenomena. And so this is as you can see here, the area explored with fMRI runs deeper than the area covers practically, in principle, the whole brain, but in this particular experiment with visual stimulation, all the occipital cortex, but not the procedures used with FNIRS. But the correlation between the 2 measures is very strong, and ideally they show very similar colocalization. This said, a lot of our work in our lab has been devoted to study neural activity directly with optical imaging, what we call the fast signal or the event-related optical signal. Now, to understand this fast optical signal refers to phenomena that are very rapid and really relate to brain activity, which is generated by external stimuli or can be generated by internal stimuli very rapidly or in preparation of responses. And so this occurs in the order of tens or hundreds of milliseconds. And one reason why we are getting this particular response is because when we have neural activity, there is a change in the conformation of the neurons themselves. So when this is showing a figure showing an image obtained with a particular individual neurons with in a preparation, and these neurons is made to be active by and to produce some form of depolarization and then hyperpolarization. And you will notice that at the moment, at the moment which we have the depolarization, we have an increase in volume of these neurons. It becomes bigger, and these neurons, this increase in volume changes the ability of the neuron to scatter light, because all the membranes here are sort of distended, and the light gets through in an easier fashion through the neuron itself. Now, we can study this phenomenon non-invasively if it happens in a lot of neurons at the same time that are concurrently depolarized, or if we use techniques that are sensitive to these changes in the transparency of the neurons in the cortex. One way of doing that is measuring the in-time interval, that is the delay that is imposed by the scattering process to the photons that travel between the source located in at one point of the head and the detector at another point of the brain. So this time delay is of the order of picoseconds, and we have technologies to measure this type of intervals. And what happens is that if the photons can travel deeper into the tissue, the deeper they can travel, the longer will be that the time it takes to move between the source and detector. So this increase in time that is obtained by increase in reduction in scattering is what is our signal. So in this, we have shown that this study, these in fact measures can be actually used to decompose time course of activity in different cortical areas. So in this image, we show this slide. I showed typical responses during visual stimulation, and on the top I have the measures obtained with near-infrared spectroscopy, and which, as I said, similar to what we would obtain with MRI. With both signal and MRI. And this indicates a time course of responses of the order of about 10-20 seconds during this period of visual stimulations. And there are also broad areas of activation during the visual stimulus that are obtained. In the same time, we can also measure individual stimuli, responsive individual stimuli, as we see these serial stimulations. And these responsive individual stimulus will show a very different pattern. They will show that some areas of the brain are activated very early, at a latency of about 50 to 100 milliseconds, and some areas of the brain are activated at much longer latency, like a couple of 100 milliseconds. And so this gives the way of sort of decomposing the brain activity that we observe in these situations between fast and slow activity. This is an example here from a study in our lab in which we have a stimulus which is occurring, which is a checkerboard and which reverses at time 0. And you will see that this animation will give you the responses to various intervals. And so here we will see that after 50 milliseconds, we have activation in medial occipital areas, which are where the signals first arrives in the cortex. And then this signal becomes more lateralized to lateral occipital areas which are presumably receive this feed forward information from the periphery at a longer delay. We can use this type of logic to study the timing and of phenomena in the brain related to many different processes. For instance, an example is this study we have done with Joe Toscano, was a Beckman fellow here. And we talked about these experiments. We studied the activity generated by ambiguous phonemes or sounds. Like, for instance, the sounds T or P, the word teach or peach. And these two sounds are in reality very similar to each other. The basic difference between the two is a particular parameter called voice onset time. which makes a particular person's perceive the letter as a T or a P. And what we find is that in certain part of the regions of the brain, early on after activation, these if we vary this voice onset time systematically between the point in which a person perceives T or P, then you will see a monotonic change in the response in these particular areas of the brain, in the amplitude of this response, depending on whether the sound is perceived as a T or a P. Notice that more precisely is the voice onset time that is important. So these particular areas are sort of an analogic representation of the sound itself. If we instead go to other regions of the auditory system that are sort of further down the line in processing, like inferior frontal gyrus instead of superior temporal gyrus areas, and they go at longer latencies, we'll find that the responses there are particularly important for the boundary level in which people change the way their perceptions from a T and a P. So in this demonstrates that the sounds are first perceived by early visual auditory areas as in an analogic fashion, but then they are coded into a more discrete sounds at a higher level of processing. We have also used this for a lot of our work has been on cognitive control and how we operate and change our way of processing stimuli depending on what is the conditions that we have. One type of task that can generate this type of cognitive control is called task switching, which we have subjects are performing a classification of stimuli according to a certain rule on a certain trial and other rules on other trials. We do this change the rules or communicate the rules with the subject by presenting a cue, and 2 seconds later we present a stimulus that need to be processed in a form or another depending on what particular rules we are having for a particular trial. And we can repeat it again and again, and maybe 2 consecutive trials we represent the same rule, and some trials we change the rule. Whenever we change a rule, something in the brain must be set up to actually accompany this change in rule, and these and make it functional. And so by comparing switch and no switch trials, we can look at the conditions in which the rules is maintained or the rules is changed. We did do this in many different paradigms, in at least 4 different paradigms here. And in each of these paradigms have different rules and a different type of cues that will indicate what particular rule is to be changed and so on. And what we find is that there are 2 types of brain responses that can be obtained. So first of all, we look at the response to the cue. And with the responses to this cue, there are depend on basically can be classified in 2 types of brain responses. Those that are we call task general, which occur more or less similarly, independent of which particular cue or task you have to switch between. And these type of activities are typically this type of situation is characterized by activity in middle frontal gyrus in the left hemisphere at a latency between 2 and 300 milliseconds. There is also a further response at about 300 to 500 milliseconds in parietal cortex. And so this sequence of events is present on all of these different tasks exactly the same way. Then there are particular type of activities, which is really very specific to what type of particular task we have. And these areas, instead of in these cases to be the response to be localized to these frontal and parietal regions we talked before, we have responses in those areas of the brain that are more closely related to the processing of the stimuli themselves. So these can be activities in our visual cortex if the rules attention is paid to visual stimuli, or auditory cortex if you pay attention to auditory stimuli, or so on and so forth. So basically we have areas that control attention and areas where attention is established. Now, this type of activity, this type of studies, we are all based on this event-related activity which is generated in the brain. But the brain itself is, as we know, is always active, and it not only is always active, but its activity is characterized by activity at various different frequencies. And we can also study these activities, these sort of different frequencies using optical imaging. and look at the similar type of bands that are used for electroencephalography or EEG bands, like those for alpha, beta, which are relatively high frequency, theta, delta, lower frequencies, and so on. So in this data refers to a study in which we studied alpha activity that is related to whether that predicts whether a particular item is going to be detected or not detected. And this activity can be observed using optical imaging in areas of in precuneus region. And this particular activity shows that this is the particular areas that are activated in this particular task. We can also, in fact, show that these areas, this activity is controlled by other activities, or at least is predicted by other activity that occurs in other regions of the brain, in frontal and parietal regions of the brain, which will mostly influence how this back portion of the cortex can gate information using this alpha phenomenon mechanism. We can also study fast optical oscillations during a working memory task, for instance, and try to determine whether they are related or not to memory function. Here we have data that we have recently obtained on a memory search task, and which we study subjects are instructed to memorize a set of stimuli, then there is a time interval, and then they are tested if one item that is part of this set of stimuli or not. We are interested here in time frequency analysis, meaning analysis of the activity fractionated by different frequencies. And what we see in this data here are complex plots indicating the locations of the brain where this activity occurs. The frequency of the activity is in different plots here. So this will be 4 Hz and this will be 18 Hz. And these areas indicated in black are areas that are activated in bold signal fMRI studies in the same paradigm. The color of these activation areas indicates the latency at which the activity occurs. And the deeper the color, the stronger the color, the more confident we are in our measurement of this activity. And this data indicate that some activities are activated earlier, and some areas are activated earlier, producing like 4 Hz activity, for instance. Some are activated later, like this one here in the medial frontal gyrus, and frontal areas. And some other areas are activated at 4 Hz, and some are activated at high frequency, like 18 Hz. We also can show that the time course of activity in these different regions, and show which areas are activated early and which are activated late. And the data show that, in fact, these sort of 4 Hz activity tend to occur early in the brain, and the 18 Hz activity occurs late during the period in which memory needs to be maintained. So this is consistent with a theory we have proposed recently, that maintenance of information in the brain is obtained to this type of frequency activity. And notice also that so we can distinguish when and where which this activity occurs, and so on. In addition, EROS is also interesting because it can be combined with a lot of different measures. And this happens because with optical imaging, we can use optic fibers to link the observations. And therefore, we can reduce the interference with other measures. So this is an example of what we can do when with this approach, by studying activity which occurs under a TMS, a transcranial magnetic stimulation pulse activation. And this graph indicates that during a transcranial magnetic stimulation pulse, you have activation of the activity in the areas under the pulse, as you might expect, as is observed with these measures. But also, interestingly enough, with some latency of about 40–50 milliseconds, you have secondary responses in other areas of the brain that are presumably activated as a function of an effective connectivity between this particular region of the brain and this other particular region of the brain. And this interval may be related to the synaptic transmission of information between one region to the other. We also recently completed a study published with human brain mapping with Matt Moore, another Beckman fellow, and who is currently a and then in the Florin Dolkos group. And this is combining optical EROS, the ventilated optical imaging, functional MRI and ERPs measured all at the same time inside the magnet, which is a relatively complicated technology to implement, mostly because combining ERPs and fMRI is quite difficult, but also we need space for the optical fibers inside the MRI coil. This said, the measurements provide a good correspondence in space between the optical and the fMRI response, as in this case from the frontal cortex, and in time of the responses with the ERP responses. There is also clearly a manipulation here of what type of response is generated in these two particular regions, ventral and parietal cortex, and this is also reflected in the optical imaging responses. So basically, we can use optical imaging system to measures to study all these different compartments of the neurovascular system, and we can study them, in fact, simultaneously and in parallel. And we can also combine these optical measures with other measures that we can obtain. And this is a group of people that actually have worked on this, that are currently working in our lab. So these are the figures that are shown on the left. Of course, Monica is my main collaborator, and Kathy is also a collaborator of everything that we do in our lab. And another person who has contributed a lot to our optical imaging technology is Antonello Chiarelli, who was a postdoc with us and is now in Italy as an assistant professor, and Ed Macklin, who has worked in our lab for a long time. I also indicate here in yellow other individuals in Beckman that have actually collaborated in this optical imaging projects that we have described, or at least those that are current collaborators. There are also others that by now have retired or moved elsewhere. And this is also other former students and postdocs. And I'm talking about our funding agency that funded this work. Besides our current grants with NIA and NIMH and DARPA, we just started a new set of collaborations with another company called Mind Portal. And this completes my talk, and I'm now would like to thank you for your attention and respond to your questions. I don't see any question in the chat. Are there any questions for me?
Oh, hey, everybody. I just allowed everybody to unmute themselves.
Thank you. I couldn't unmute myself.
Yes. Yeah. So yeah, Stacy was the host, and she just came in here to tell me that her computer seized up and she couldn't let people unmute. But I just did that because I'm the co-host. So now you guys are all okay to unmute and engage in conversation. Thanks.
Thanks so much, Patty. Let me see. Thank you, Gabriele. That was a wonderful talk. It was beautifully presented, and I certainly learned a lot. I have a lot of questions myself, and I would encourage others to unmute yourself and ask questions, or put them in the chat if you'd like.
Looks like Brad is our first question.
Great. Yep. I will, and I'll simplify here a little bit, make sure there's enough time for other people. You show big effect of fitness and the aging group splitting so big into two groups, one where the signal completely gone, maybe even negative, versus the high fitness group where they match the young people very well. Do you think there are clinical conditions to fitness, obviously mediating the vascular properties, but I'm wondering about clinical conditions and the increased use of certain pharmaceuticals and like the focus on cholesterol and things like that going forward. Do you think that some of those physiological components will show up in the measure you're making, or, I mean, obviously fitness is the best way to go. It hits all these multifactorial pieces, but I was just wondering what your thoughts were on that.
Yes, first of all, I need to explain that in that particular experiment, we had sort of extreme groups in terms of physical fitness. So we recruited individuals that were in the top third, in the bottom third of fitness, in cardiorespiratory fitness, according to some type of previous data that, in this case, our collaborator in kinesiology had set up. This said that, yes, I believe that there certain type of interventions actually change the response, the cerebrovascular response. We know very well that, for instance, I presented data here about the fact that cocoflavanols have a very rapid effect on cerebrovascular reactivity. And in fact, there is a phenomenon which occurs within an hour from when you take these cocoflavanols. Some people also believe that cocoflavanols have a long-term effects, and that they will actually improve your cerebrovascular activity and generally cerebrovascular function over a very extended period of time. And there are some data that in support of this idea. So, but this is an example of just one manipulation, which is, in a sense, it's a pharmacological manipulation, but it's really also a dietary manipulation. Does it address your question?
Yeah, to follow up, I'm curious, so can you do the diffuse optical measurements while a person is vigorously exercising?
Yes and no. Okay, in principle, it should be doable. In practice, there is a possibility of artifacts created by the coupling of the measurements with the brain, and also what happens in the very superficial areas of the head during a particular exercise. So one needs to be very careful about this. My answer to this question is that in principle, yes, but it's not so simple. And so it will require presumably very good control of movement artifacts, as well as the coupling of the instrument or the measurements instruments with the head.
And well, I'll keep asking questions until someone raises their hand, because I was curious to know whether or not neurovascular health is heterogeneous within a given individual. In other words, are there regions of the brain that are healthier than others, or is it pretty much a single individual is just going to have the same kind of state of vascular health? Actually,
that is one of the graphs that I presented would be answering your question, addressing your question. So we were actually interested exactly in that question. And this is why we did this type of analysis that I presented here. I don't know if I went through very quickly, so I'm not sure how clear it was. So when we studied the pulse relaxation function, which is basically a measure of physical health, of arterial health, we find that this parameter is, you can measure these parameters separately in all different parts of the brain. So this is one thing we can do in our system. And let's see if I can fix this. Okay, so these measures on the left indicates within individual correlation between pulse relaxation function in a particular region of the brain. Here there are about 50 regions of the brains that are separated from each other using the Desikan Kiliani atlas. And these different regions are quantified. For these regions, we quantify the pulse, the arterial pulse, and the volume of those regions. Then we actually, for each of these regions, we determine whether these individuals have more of it or less of it relative to the other individuals of this particular region, and had a bigger or smaller pulse relaxation function in that region. And then we looked at whether there is a correlation between a subject having higher value of pulse relaxation function in one region, and are they the same subject that have a bigger volume in that region? And those that have lower, Okay. In this way, we are basically creating an analysis in which we are, all of these are standardized, and they are just variations of the regions relative to the other regions for each individual. So there is a correlation within individual of those areas in which they are better than the norm, and those areas that are worse than the norm in pulse relaxation function, are they bigger volume than the norm or smaller volume than the norm in the same individuals? And there is in fact a positive correlation between that. And this is what this data indicates. Now, this is only evident in older individuals. In younger individuals, the fact that you have arterial function, good or bad, doesn't affect much about the fact that whether that particular region is bigger or smaller than it would be expected to be. And because these two is the size of that is not due just to arterial function. People are born with many genetic factors that, or other phenomena or other things happen in their life that will make a particular region of the brain bigger in that individuals rather than another. But after age 48, the pulse, the arterial function becomes a major determinant in what actually guides whether this particular region will become smaller or larger. Does he address your question?
Yes, thank you.
So in this sense, it is actually a regional phenomenon. It is not true that that these will occur equally across the whole brain. Some regions will be affected more. We are, in the current study, in the current grant, we are interested in knowing what controls this. And that we also have the two sorts of models of what happens, is that there are some regions that tend to be those that show the phenomena more than others. And there can be therefore sort of weak regions that tend to be those in which the problem are more likely to occur, or vice versa. There are different patterns for different individuals, and this may be due to phenomena that are difficult to at this moment to identify. But yes.
Let me ask if there are other questions. I certainly still have some. So, but I don't want to take up all of the time to allow others to ask.
Yeah, whatever you prefer, Jeff.
All right, I will keep firing away my questions then. I'm curious about the voice onset time, and I have heard, and I don't, I have no idea if it's true or not, but I've heard that one way to help remember names when you're being introduced to people is to be paying very close attention to the rise and fall of the audio signal as the name is being announced. I'm curious if you can then relate sort of this stimulus of the onset time to memory. I mean, whether or not this, to sort of test this idea, maybe it's been tested and maybe that's where it comes from, or maybe, I heard something that's just completely false. I don't know. I'm just curious if essentially a way to track those relationships.
Well, there can be physical characteristics of a particular stimulus that sometimes may help memorizing and distinguishing from other stimuli. So from a psychological point of view, it is the stimuli may be more or less confusable with each other. And the more confusable they are with each other, the more difficult it is to form a distinctive link between a particular stimulus and other stimuli. So you can make stimuli more or less distinctive in many ways. For instance, you can make them pay attention more to certain particular physical properties of a stimulus, or you can make them distinctive in other ways or manners. But distinctiveness is a very important characteristic, a basic characteristics of memory. So there is a lot of research starting in the 1930s by people in the Gestalt psychology that sort of demonstrated that stimuli that are more distinctive are much more memorable. In fact, actually, it is a common way in which advertising is done. in which you tend to try to make whatever you have very highly distinctive. And creating this distinctiveness, you make things much more memorable. So there is an old tradition in psychology of at least 80 years of research indicating the distinctive items. And we can also, this is actually work that Monica has done in person, show that you can predict whether a stimulus will be memorized or not by how the brain responds to it the first time. And in particular, by how the brain notices distinctiveness in the stimulus. And so even if you don't manipulate the distinctiveness, but you just measure the distinctiveness, the subjective distinctiveness of the for each individuals, you still get differences of this type. Well, this goes back to when Monica did her dissertation work.
Well, I have two more questions, but we're already past the hour, so I'll have to wait and ask you the next time I see you in person.
Okay. Thank you.
I'm curious so much about what I heard, and it's a really beautiful presentation that describes the interaction of light with matter to really understand what's going on as we try to understand how our brain processing and perception all works together. So it was extremely well delivered. Thank you so much, Gabriele. I really appreciate hearing your talk today.
Thank you.
All right, we have one more director's seminar. It'll be in about, I think it'll be in about a month. So please join us then, and thank you.
Bye.